cartilage. These bones which get modeled from cartilage. All right. So we have here a baby at 8 weeks, a fetus at 8 weeks old, okay, in the mother's uterus. This is when the ossification of the bones start. First, there is going to be a cartilage model, all right? Now, when there is a cartilage model, with time, the perichondrium, remember that lining of cartilage, the perichondrium is going to be invaded with bone-forming cells. It's going to be invaded with osteoblasts. These osteoblasts are going to start depositing bone tissue. They're going to start depositing osteoid. Once osteoid is deposited here, there will be death of the chondrocytes because the chondrocytes will now be deprived of nutrients from the bloodstream. Okay, they'll be deprived of nutrients from the bloodstream. So slowly, bit by bit, the chondrocytes will do what? Will start dying. And therefore, the bone matrix will start increasing. As bone matrix is increasing, the cells that are producing the bone matrix are slowly also growing and being trapped. But remember, that bone collar there, as there is deposition of the matrix, it also changes from perichondrium and becomes periosteum. Meaning in the periosteum, remember we have the inner layer, which is osteogenic, okay, which contains the precursor cells for bone, okay? So those precursor cells will continue becoming osteoblasts, and with time, the osteoblasts will continue becoming osteocytes, and with time, though those lamellae will form or the lacuna will form, all right? Now, as time goes on, we are going to find that in the middle of the diaphysis, we are going to find the appearance of what is known as the primary ossification center, where bone matrix is also going to start appearing and bone starts hardening in the middle. And that is what is known as the primary ossification center. And also there in the middle, there is going to be death of these, of the chondrocytes that are there. And with time, there's going to just be a space there. Now, as that happens, there's going to be invasion of blood vessels now. The blood vessels will invade the periosteum. And once they invade the periosteum, they are going to go in and more cells are going to form. There is going to be formation of the endosteum inside, covering the inner lining of bone, okay? And then with time, at about the time of the birth of the child, of this baby, we're going to see an appearance of the secondary ossification center at the epiphyses. We're going to see an appearance of the secondary ossification center at the epiphyses. And the same thing is going to happen. The only difference is that here in the epiphyses now, we're going to see the appearance of spongy bone, okay, this or trabecular bone. And the other difference is that the hyaline cartilage will not completely finish when it comes to the epiphyses. So there will be a remainder of hyaline cartilage at the top of the epiphyses, which is going to be the articular cartilage, okay? And in the secondary ossification center, also we are going to see the appearance of blood vessels, which are going to start bringing nutrients now to the osteocytes that are there. As this person is growing, remember we talked about cartilage not completely ossifying at birth. we remain with a certain layer of cartilage, which is going to be known as an epiphyseal growth plate. That epiphyseal growth plate allows for the elongation of bones, okay? Allows for the elongation of bones, which is why when you go for x-ray and you have said, I am 13, when you are actually 28, they will know that you are not 13 because you won't have an epiphyseal growth plate. So this picture is an x-ray of two people with different ages, okay? This one on A is a child. This one is an adult. If you look at this adult, you are not going to see an epiphyseal growth plate. You only see a line where the growth plate was. So we said that all these bones, the bones of the wrist, are phalanges, and all the phalanges are long bones because why? They have epiphyses, epiphysis, and diaphysis. Okay, so all these long bones, you find that this person who is an adult, they don't have an epiphysis growth plate. However, when you look at this one who is a child, you can see cartilage, you cannot see it on x-ray, and therefore you are seeing that there are places there where there is cartilage, and you know this one is a child. And that is how they're going to know us. We go to the word and we say, no, me, I'm 15, and yet I'm 20 shanyuko. And they're going to know that this person is lying, like our footballers. Footballer who says, no, me, I was born in 1986. My mother died in 1984. It's crazy. Anyway, now we have intramembranous ossification. Intramembranous ossification is where now bones will ossify from a membrane that is derived from mesenchyme. Okay, a membrane that is derived from mesenchyme. Here, like we said, mesenchymal cells have the ability to just move, and then they start differentiating and they become another type of cell. Okay, so we're going to find membranes like this one under the skin, and they're going to be there's going to be deposition of mesenchymal cells, which will start producing extracellular matrix of bone, which is known as osteoid. Okay, once there is deposition of osteoid selected, so this area is selected centrally, okay, and the mesenchymal cells cluster and differentiate into osteoblasts, okay, forming an ossification center right in the middle of the membrane. So the bone matrix, which is the osteoid, is secreted within the fibrous membrane. So you've seen the bone matrix is being secreted. As it is being secreted, some of the cells are being trapped, okay? And as they are trapped, they are maturing, they are now becoming osteocytes. On the outside still, we're going to have osteoblasts, which are maturing and still producing osteoid. Osteoblasts begin to secrete osteoid, which is mineralized within a few days, mineralized with those minerals we talked about, calcium, phosphate, okay? And as the minerals are trapped there, it means that the bone is now starting to become hard. It's now becoming ossified, okay? So there is now formation of woven bone inside, okay? And there is formation of periosteum around it, okay? If you look at those cells, they're about to start forming periosteum around it. Accumulating osteoid is laid down between embryonic blood vessels, okay? And these form a random network within the meshwork of the woven bone. So this is where we're going to find that inside there's going to be uh, a network. Inside the flat bones, there's going to be a network of bony trabeculae instead of lamella. Okay. There's vascularization. So vascularized mesenchyme condenses on the external surface of the woven bone and it becomes the periosteum. Like I was saying, these small cells around here, they condense around this bone and become periosteum. Bone collar of compact bone forms and red marrow appears. So this is why you're going to find in flat bones, you have red marrow, but no yellow marrow because we don't even have a canal. The spongy bone, like we said earlier, is also known as the diploid. It's also known as diploid. Okay. Now we have factors regulating bone growth. There are some people whose bones don't grow so much and so they end up being short. Okay. So we have things that will regulate all those things. For example, things like vitamin D. Vitamin D is one of the vitamins that, one of the fat soluble vitamins that increases the absorption of calcium from the GIT. As you eat, vitamins, the availability of vitamin D will cause absorption of calcium from the GIT into the blood, meaning it can go and get deposited in the bone. And then we have parathormone, which is a hormone from the parathyroid glands. Okay, now we said that the parathormone and calcitonin are antagonistic towards each other, meaning they do the opposite. So parathyroid hormone is that hormone which works hand in hand with the osteoclasts so that when there is too much calcium deposited on the bone, it would be removed from the bone and it will be taken into the bloodstream and calcium in the blood will do what? Will increase. Or if you do not have enough calcium in the blood, parathormone will be released so that bones can release some of the calcium that it has stored. Calcitonin, on the other hand, encourages calcium to be deposited on the bones. So these two hormones they work antagonistic to each other. And so you're going to find that the two hormones are factors also that regulate the growth of bone. Growth hormone and thyroid hormone. These also regulate the growth of a person in general, as well as how much bone grows. Sex hormones, such as those hormones produced during adolescence, and you find that this person now grows, there's a growth spurt because of those sex hormones. And so males become tall, very much taller. You'd find that you were growing up with this male nicely, he was nicely shorter than you. But when you reach puberty, suddenly he becomes much taller than you because the hormones will work on bones in different places of this human being. Okay, so on females, they will work on things like the hip bones so that your hip bones can widen, okay, and stuff like that. Okay, so like I was saying, this is just a summary slide. We have bone remodeling. So this is carried out by the osteoclasts, which will resorb the extra bone, the osteoblasts, which will deposit bone tissue. And this remodeling is triggered by remodeling, by the way, can be in any way, it can be at any time. And even during healing, bone healing, as your bone is healing, you find that there may be too much deposition of calcium at a particular place as the bone is healing, And so the bone is supposed to remodel itself nicely. Okay. And so those are the things that um, we were talking about. All right. Any questions?
No questions for now.
Okay. But you guys don't ask any questions. Even from the videos that I post, I even wonder if you would listen to those. I hope you do.
The questions will start coming soon, madam.
That's coming. Okay. We'll be waiting. So we quickly look at an introduction of blood. We can quickly introduce blood so that next time we meet, we just conclude. We can quickly introduce blood so that next time we meet, we can just conclude. So we have blood. We said that blood is also is also one of the specialized connective tissues. Okay, so blood is a connective tissue. It is composed of plasma and formed elements, physical formed elements. And some of the physically formed elements include the three blood cells, the erythrocytes, the leukocytes, and the platelets. Okay, the erythrocytes, the leukocytes, and the platelets. So at the end of the session, you people should be able to explain blood, what the blood is exactly, what is composed of blood, what are the functions of blood. Should be able to talk about the clotting mechanism, the blood groups, and the antigen antibody reactions. So we are saying that blood makes up 7% of the body weight. It carries oxygen, nutrients, hormones, heat, antibodies, and clotting factors. So it will carry oxygen from the lungs to the tissues, carbon dioxide from the tissues to the lungs, nutrients from the GIT to all body parts, waste products from all body parts to where they're supposed to be excreted, hormones from endocrine glands to the organs that they're targeting or the glands that they're targeting. They carry heat, which is produced inside the body in metabolically active organs, and they go and provide the heat to other organs that are not metabolically active. Okay, they carry antibodies from one place to the other. They also carry clotting factors from where they are produced, especially from the liver, and they carry them to where they are needed. Plasma. Plasma is an aqueous solution and the pH is about 7.4. It constitutes about 55% of all the blood. Dissolved components are mostly plasma proteins, okay? But they may also include nutrients, gases, waste products, hormones, ions, and these things are collectively known as electrolytes. The major plasma proteins include albumin. I think we know the function of albumin, the one that controls the oncotic and osmotic pressures, which means it controls how much fluid goes into and outside, how much fluid goes into the intravascular spaces and how much fluid goes out of the extravascular spaces. So it's a very, very important protein. If you don't have this protein, you'll find yourself having edema because there will be no control of which fluid should stay in the blood vessels, and the fluid will be moving from the intravascular spaces to the extravascular spaces. There is no control of osmotic pressure. Then we have A globulins and B globulins, alpha and beta globulins. And these include transferrin, fibronectin, prothrombin, lipoproteins. And then we have immunoglobulins, which are part of the F globulins. And then we have fibrinogen, In fact, apart from fibrinogen, we have other clotting factors also suspended in protein. When you look at blood as a whole, this is what is formed in blood. We have the plasma part and the formed elements. The formed elements are these ones and these ones. When you put this blood in a test tube that has nothing in it, those plain top test tubes, those are red. They are the plain test tubes. They are red topped and yellow, right?
Right. Yes, madam.
Mm. So you put this blood in here, you're going to find that red blood cells are going to settle at the bottom, and this is what is known as hematocrit. And then we're going to find there is a coat on just on top of the red blood cells that appears whitish. That is where the white blood cells and the platelets will be. And then we have this other part of plasma. And these tables are showing us the constituents of plasma. Okay, we have albumins, globulins, fibrinogen, and other proteins. Okay, all these. And then we have these white blood cells that are going to be found here, as well as the platelets that are going to be found here. This is known as the Buffy coat. This is known as the hematocrit. This is known as the plasma. So that is what we're talking about. So the erythrocytes now. Okay, erythrocytes, I think at this level, we all know that erythrocytes, one, they don't have any nuclei, but they are filled with hemoglobin. They are filled with hemoglobin. They don't have any organelles so that they provide enough hemoglobin to attach to oxygen and carbon dioxide. Okay, so they are very flexible. They can move even in very tiny blood vessels. They do not move from blood to the external, to the extravascular spaces. They are the only cells that will stick to inside the intravascular spaces. They never go out. Their lifespan is 120. Is 120. Um, their lifespan is 120 days. Okay, and so when they reach that, they have the ability to form this, which is called rolu. Rolu, okay? They aggregate together. When they reach a small blood vessel, they find a way of aggregating together and moving in that blood vessel. And that is what is known as a rolu, that aggregate of the red blood cells. Okay, that is what that is called. And then we have leukocytes. These are the white blood cells. And we know that white blood cells are of two groups. And these two groups are grouped, one according to the shape of their nuclei, two according to the granules that are found in them. So we have one group, the granulocytes, and these granulocytes are represented in this picture by these diagrams. So we have these granulocytes, we have the neutrophil. By the way, one of the ways I managed to remember is when you say all the granulocytes, all the granulocytes have fill at the end. All the granulocytes have fill at the end. Okay. Sorry, just a minute. Hello. Okay, so all the granulocytes have fill at the end. So you have neutrophils, eosinophils, basophils. These are granulocytes. And then we have the agranulocytes, the lymphocytes. These lymphocytes, but we all know that lymphocytes are grouped into two. We have the B lymphocyte and the T lymphocytes. They are both produced in the bone marrow. We'll look at uh, blood formation very soon, maybe not today. Then we have the monocytes. these lymphocytes are divided into B lymphocytes and T lymphocytes. They are both produced in the bone marrow, but the B lymphocytes will mature in the bone marrow and the T lymphocytes will mature in the thymus gland. We have monocytes which mature into different types of cells, for example, macrophages, and then macrophages will move from the bloodstream and go to different organs and will be known by a different name in each organ. Okay, so these are the two groups of the white blood cells. All right, those are the two groups of the white. The granulocytes have a lifespan of 1 to only a few days, and after those few days, they die nicely by apoptosis. Apoptosis is programmed cell death, and then when they die, they are phagocytosed by the macrophages in the organs where they die from. They have polymorphic nuclei. You're going to notice that the granulocytes will have nuclei with more than 2 or more lobes. Okay, so you're going to see that one, there's going to be a lot of granules in them, but inside, when you look at the nucleus, it will look like there is either 2 nuclei or 3, but it is just one with the number of lobes. Okay, and that is what one of the other things that make it different from the granulocytes. Okay, and then we have the granulocytes with no granules. The nuclei is one. Obviously, some of them, they will have a very large nucleus. Okay, very large nucleus, but it is just one. There are no granules. Okay, they help, like all the white blood cells, as we know, they help in fighting off microorganisms. Okay, they fight off microorganisms. So we have different white blood cells, and these different white blood cells have different functions. Like we said, we have those neutrophils, which are granulocytes. All these three here on this slide are granulocytes. These granulocytes are neutrophils, which engulf microorganisms by phagocytosis. We have eosinophils, which are specialized in inflammatory reactions, okay? And then we have basophils. In fact, eosinophils in allergic inflammatory reactions, okay? Not just any other type of inflammatory reactions, allergic inflammatory reactions. And then we have basophils. And the basophils contain cytoplasmic granules, and these are the ones that contain things like heparin, which is an anticoagulant, histamine, which is an inflammatory product. So all these, they fight against infection. Then we have, these are granulocytes now. The first granulocytes we'll look at are the monocytes. These circulate in the blood, okay? They are phagocytic, okay? However, some of them will migrate into different tissues. Different tissues, they'll be known by different names. For example, in the central nervous system, they'll be known as microglia. For example, in the liver, they'll be known as Kupffer cells. For example, in the umbilical cord, they'll be known as half-Bauer cells. Okay, so they are known by different names depending on where they are. Then we have lymphocytes, smaller than monocytes, large nuclei. They circulate in blood, present in great numbers in lymphatic tissue. We know that in lymphatic tissue, we are going to find a lot of these lymphocytes, both the B and the T lymphocytes. These ones, they respond to antigens, okay? So the cells are regarded, by lymphocytes as abnormal, e.g. those that have been invaded by macrophages. Those ones, they'll also be attacked by the lymphocytes, okay? Other things that lymphocytes respond to include pollen, fungi, bacteria, and then we said that there are two distinct types of lymphocytes. We have T lymphocytes and B lymphocytes. Okay, we have T lymphocytes and B lymphocytes. Okay, so the last slide for today, we are looking at the platelets. These are not cells, they are particles, they are fragments which are found in blood plasma, but they are very important in blood clotting. They are very tiny, minute particles, okay? Very minute particles measuring 2 to 4 micrometers in diameter, okay? The platelets have a lifespan of 10 days, okay? So for now, we'll end here until I finish making the notes on blood. Then I'll send the notes for connective tissue tonight. Then we'll look at, we'll continue blood the next time we meet. Any questions?
Madam,
Mm-hmm.
just a request on the recordings. Some of them are a bit noisy. So it's hard to, yes, it's hard to understand some things or to hear what is being said.
Okay, so we should stop the recordings or maybe...
Or no, maybe just getting them more clear.
Okay.
Yes, yes, but they are very helpful.
Okay, no problem. Okay, have a good night.
Good night, madam. For me, I'm very happy today. It's one of my classes. At least about 10% managed to get your voice. Others mostly I get your voice from the recordings I failed to go through my classroom.
You failed to get the recordings completely.
No, I'm saying the recordings. I'm saying I usually get your voice or your lectures through the recordings. When it's currently happening, I usually fail to log in. So today is one of the days I've managed to attend it 10%, getting your voice. Otherwise, I just see maybe the writings and everything.